Okay, so catabolic reactions are reactions that break things down, such as the reaction of cellular respiration. It's responsible for breaking food down, for breaking glucose down into ATP, and as long as oxygen is present, that's where you get a lot of your ATP when oxygen is present, when you're actually inhaling O2. And if you want to build something, like maybe build a muscle or build complex macromolecules, you have to go with the anabolic pathways. Okay, so for example, if you want to build an amino acid from simple elements, or if you want to build a protein from amino acids, all of that is called anabolic reactions. So they will simply go opposite to each other, and ultimately the catabolic reactions will provide energy for the anabolic reactions. So to build something, you must have energy in place. To get that energy in place, you must have ATP available through catabolic reactions. O2 is going to be a major reactant for you. You must always be inhaling O2 to actually get the ATP that your body needs. Now, there is something called the change in free energy, or delta G, which can tell you if the reaction is going to be giving energy out or taking energy. For example, the reaction of cellular respiration, which is what every single cell should be able to do to give ATP to itself. This reaction will be giving you ATP. And if it gives you ATP, it's called exergonic in biology. Okay, it's giving you energy. It gives out energy, basically. But photosynthesis needs energy of the sun to do its job. So because you have to have an external source of energy to do the job, that will be called endergonic reaction. How does that relate to delta G? Delta G is going to be negative for exergonic reaction, but it's going to be positive for endergonic reaction. So the sign of delta G matters a lot here, basically. So energy is released when the reaction is exergonic, when delta G is less than 0. Energy is released like the case with cellular respiration. To give us lots of ATP. But when energy is absorbed, like the case of photosynthesis, that's when delta G has to be more than 0. Okay. So for cellular respiration, delta G is negative. But for photosynthesis, delta G is the same exact number, but positive. Okay. So making sugar happens during photosynthesis, which basically means that making sugar has a positive delta G. ATP is going to be of great focus in this chapter because of the whole point of it storing energy. So what's ATP, or adenosine triphosphate, exactly made of? It's made of a sugar called the ribose sugar, the same sugar that we saw before in RNA. It's made of a nitrogenous base called adenine, which is found in both DNA and RNA, along with 3 phosphate groups, hence the name triphosphate. Okay. Now, you can break one of the phosphates away by water, by adding water in a process called ATP hydrolysis. And once you've done that, you will now release energy. And releasing energy is called exergonic, hence the delta G being of a negative sign. Here is what ATP looks like, adenosine triphosphate. It's made of adenine, which has, as you guys can see, 2 rings. It's made of ribose sugar, which, as you guys can see, it has OH on the second carbon, different from deoxyribose sugar, which lacks that OH, along with 3 phosphate groups. And once you have released one phosphate, energy gets released to do work. What's an example of a process that occurs in your cells that requires ATP? Active transport. Active transport, which we covered back in Unit 2, is responsible for moving something from low concentration to high concentration while using ATP to do so. So the point of using ATP is because ATP has an extra P that it can give, and now when a transport protein has P on it, we call this protein phosphorylated. At this moment, this transport protein is not transporting anything. But now once the phosphate is removed, the solute will then be transported. Okay, so maybe the transport protein is only open in one way. Okay, when there is phosphate, once the P is removed, it's going to open now in the opposite way. So sometimes phosphate can be used to activate things or deactivate them. In this case, the ATP phosphorylates the transport protein, causing a shape change that allows them to transport solute. So if there is no phosphate, the transport protein is going to be unable of doing anything. When it comes to moving a vesicle, which are used to move things from the rough ER to the Golgi body, or from the Golgi body to the cell membrane, we also have to use ATP to do so. As you can see here, energy from catabolic reactions, which are called exergonic energy releasing reactions, is used to support the endergonic reactions. So ATP hydrolysis yields or provides energy, and of course you must have water with it to do so, to break one phosphate away. We can also get ATP back by simply recombining ADP with P to get ATP back. And this process is not going to give you energy. It requires energy to happen. That's why it's going to be a positive delta G, which means endergonic reaction. Endergonic reaction means a positive delta G. Energy is required to do something. See, free energy must be spent to make this process occur. Catabolic reactions provide the energy for anabolic reactions. An example of a catabolic reaction is going to be the process of cellular respiration. Enzymes can speed up chemical reactions, but they do so with a limit. They can only do so if they are provided the vitamins that they require. You must always have vitamins or minerals in your diet to make the enzymes do better. How will enzymes make the reactions faster? They will lower the activation energy. The activation energy is responsible for keeping the reaction not happening or taking lots of energy to happen. So by lowering it, the reaction will happen faster. Each enzyme is going to be super specific. Like to break down sucrose, you must use the enzyme sucrase along with water. And once you've broken down sucrose, you break down this bond, you will end up with an OH on one side and H on the other side, because the water will contribute an OH to one side and also contribute an H to the other side. And you will get glucose and fructose by breaking sucrose down. So an enzyme is going to be essentially a protein. Just like a protein, enzymes are going to be made of amino acids, and it will act as a catalyst to speed chemical reactions up. Enzymes are not consumed by the reaction. They're not consumed by the reaction. So here we go. To go from reactants to products, you must first elevate energy-wise to transition state, which is like a thing between reactants and products. And whenever the products are at a lower energy than the reactants, that's when delta G is negative. That's when the reaction is exergonic. So exergonic reactions are those where the products are at a lower energy than the reactants. Okay, so the products have less energy than the reactants.
It's like in chemistry with the exothermic and the endothermic.
Same exact thing, but now it's called exergonic. Yes, releasing energy to be used by anabolic reactions, or maybe by you to stay warm or do work for your body. So ultimately, that's the activation energy. The higher it is, the less products we get. And the lower it is, the more products we get. And that's where the enzyme jumps in. It will make the reaction faster by lowering this activation energy. An enzyme will never change delta G. It will never make an impossible reaction possible. It will simply make the reaction faster. That's all it does. It makes the reaction faster. Every enzyme is going to be specific. Only one substance can bind to the enzyme. That's called a substrate. The substrate of one enzyme cannot bind to a different enzyme. Each enzyme has its own substrate. Once they bind to each other, you will get something called the enzyme substrate complex. And now the reaction starts to happen to make the substrate into something else. They bind to each other by something called the induced fit, which basically means that the shape doesn't have to be 100% fit, but just good enough. It's like when you buy a phone cover, you're able to, squeeze the phone in there to an extent, but it's not firm. Whenever you see ACE, that basically means enzyme. If you see ACE at the end of the word, that basically means enzyme. Like sucrase breaks sucrose down. So sucrase can only bind to sucrose, but not to maltose. So the active site is going to be the special place about the enzyme where the substrate is able to fit and bond there for a while until the reaction is finished. Now the active site of the enzyme is going to be made up by the R groups of a few amino acids. And that's how important the R groups will be to make up the composition and the reaction of the active site. So here's how it works. The substrate will be able to enter the active site. The enzyme has to change shape slightly so that its active site will be able to take the substrate in. That's called induced fit. And once that happens, you will now have interactions between the substrate and the enzyme by hydrogen bonds and also ionic bonds, all of which are facilitated by the R groups. The enzyme will then lower the activation energy, making the reaction more possible and also faster, and the substrates become a product. Products get released, and now the enzyme is going to be reusable for another reaction. Now, if you want to make the reaction faster, you can obviously add more substrate, because more substrate would mean more collisions with the enzymes, which basically means more active sites are going to be filled up. But at some point, if you have a lot of substrates, but the number of the enzymes is still low, the reaction cannot become faster anymore. Because if you have 1,000 substrates for only 20 enzymes, only 20 substrates can bind at a time, correct? So the best way to make the reaction faster is to make the enzyme concentration more. Now, how would a person do that? By taking in vitamins, right? By taking in mineral tablets. You would make the reactions in your body faster, which basically means weight loss will be easier, more energy available to you during the day, and so on. Now, the best source of the enzymes or the vitamins or the minerals is not pills, is natural fruits, natural vegetables, basically. That's how you get your good amount of vitamins and also a good amount of minerals. No pills, just fruits and vegetables. Okay, will do the job. So if all the active sites have been occupied, the enzyme becomes saturated, and the diagram for the reaction rate will be increasing initially, but then at some point it will stabilize, and that point of stabilization is the point where all the active sites have been occupied. All the active sites are occupied at that point. So the best way is to have more enzymes to make the reaction faster. If you guys are faced with a concentration of a single substance over time, what type of graph do we have to use? A bar graph or a line graph? Line graph, because you're looking at one thing over a long period of time, you have to be using a line graph, which basically means you must be connecting the dots with lines like line 1, line 2, line 3, and so on. And here, if you were to graph this, you would have no error bars because there's no plus or minus. Which variable here has to go on the x-axis? The time, because we can control it. But the concentration we don't control. It's called the dependent variable. It will go on the y-axis. Now, based on this data, do we see a positive correlation or a negative correlation between time and concentration? Positive, but at some point, more time does not result in more concentration. At some point, the enzymes have been saturated. Okay? That means their active sites have been all occupied up. What are some ways to affect enzyme activity? By affecting temperature and also pH. You see, an enzyme is going to be very sensitive, just like a protein, to those two variables. Enzymes and proteins, both of them have a 3D shape, which determines their function. And if this 3D structure is damaged by any way, the function will also be damaged. Now, of course, some conditions will be optimal, which basically means best. For example, a temperature of your body of 37 Celsius is really good. Or a pH of a 2 in the stomach is really good. Or a pH of 8 in the intestines, small intestines, is also really good. Some enzymes like the pH of 2 and some like the pH of 8. You can never force an enzyme that likes the pH of 8 to work at the pH of 2. It will simply not happen. In fact, the enzyme will lose its function, it will lose its shape, and it's going to be damaged permanently. There's no going back, basically. That's called denaturation of the enzyme. Now, when it comes to a human, the best temperature for the enzyme to work is going to be 37 Celsius. But some organisms, like thermophilic, heat-loving bacteria, would love their enzymes to work at 75 Celsius. Hence, you're going to have a peak at 75 Celsius for this bacteria versus just 37 Celsius for a human. Different organisms could lead to different optimal temperatures. And same goes for the pH. Trypsin enzyme, which specializes in the small intestine, works best at the pH of 8 versus pepsin, a stomach enzyme, works best at the pH of 2. If you want to make the enzymes work better, you have to give them help by something called a coenzyme if it's going to be organic. If it's going to be inorganic, it's simply called a mineral. So cofactors are helpful things for the enzyme. They can make the enzyme work better, and they will come in two varieties, either organic or inorganic. If they're organic, they basically both have carbon and hydrogen at the same time. They're called vitamins. But if they're inorganic, they're called minerals. Okay. Now, enzymes can also be inhibited or made to stop their work. So some things can inhibit the enzyme permanently, and some can do it only for a period of time. Both of these inhibitors, whether permanent or not, are called inhibitors. they can be competing with the enzyme, or they can be not competing with the, competing with the substrate, or they can be not competing with the substrate. You see, the substrate only has one place to go on the enzyme called the active site. So if the inhibitor competes with the substrate, it basically means it wants to also bind to the active site. But if the inhibitor does not compete with the substrate, it wants to bind to a place other than the active site. And this place will be called the allosteric site. Okay? So, let's right now make a table to compare both types of inhibitors. So, if the inhibitor is going to be competitive, it will compete with the substrate to bind to the same place, to bind to the active site. If it's not competitive, does not compete with the substrate, and it binds to another place called the allosteric site. Now, if you ever have used rat poison, it will be the second type. Rat poison is this type. It will block some enzyme responsible for a good health of the rat permanently, forever. That means one dose and it will die. But antibiotics will have to always compete with the substrate. That's why the doctor might say, you must take this twice a day, or let's say one time a day, not only one time, because it will always have to compete with the substrate. Whichever has the higher concentration wins. Okay? So basically, if it's competitive, it can be overcome by simply having more substrate. This one, it cannot be undone. That's it. Once the rat ingested the poison, it will die. Unlike antibiotics, which you must take for a course of at least 3 pills. This is an enzyme. It's got two sites, the active site and the allosteric site. Two things combine on the active site, the substrate and the competitive inhibitor. One thing combined on the allosteric site, which is non-competitive inhibitor. Okay? There is no competition with anyone. So if it binds here, it will not compete with the substrate. We just need a small amount. If it's competitive, we need a large amount, because it must now compete with the substrate.
So are we the funniest class you've had?
Now the point is, feedback inhibition is the way we can control enzyme activity. Okay? We have to control enzyme activity. We have to shut down the enzyme once it has finished its job. For example, if the enzyme's job is to make isoleucine, and now you have made enough isoleucine, do you have to keep making?
No.
No. So now the isoleucine itself, which is the end product, will then go and bind to the enzyme, to its allosteric site, and it will tell it to stop working. So if you want to stop making something, that end product itself has to go to the enzyme, binds to where? To the allosteric site, to shut down the entire process. This happens all the time in your body. Once you've made enough something, you must now stop. For example, let's say you made $1 million. Do you have to keep making more?
Yes.
No, that's it. You're done.
No.
According to biology, you're done. That's it. Relax.
Yes, master.
Let me ask you guys a question. Who has mitochondria? Animals or plants or both?
Both.
Animals.
Both actually. And who has a chloroplast? Plant or
Plant.
animal or both? Only plants. Thank you. Whenever you see this folded organelle
It's a matter.
or foldings, basically, it's going to be mitochondria.
So Loki looks like a lysosome though.
How? Look at all that folding.
Where's the lysosome?
So Loki looks like bread.
It is not shown.
On the animal cell?
What doesn't look
So
like
that's the
bread.
nucleus?
That probably is the lysosome. Probably that one.
No.
So it looks
Probably.
like bread.
No, that's a vesicle.
I'm not sure. They almost look the same.
This is
That's
the
a
lysosome.
vesicle. That looks like a vesicle
No.
right here.
These
Oh
dots.
yeah, the double membrane thing.
Yeah,
This one's the thingy.
that looks like a lysosome.
This one.
That's called the centriole
Oh.
or the centrosome. That's the mitochondria.
What's the difference between rough and smooth? Where like how can you say?
The rough one is not shown here. It must have dots to be rough.
Oh, the
Ribosomes,
ribosomes.
right?
Yeah,
Remember it.
very good. So, cellular respiration is responsible for turning glucose into ATP. And 90% of the ATP is made in the mitochondria. Now, to do that, you must have lots of O2, you know. So basically, you cannot be, let's say, holding your breath and making mitochondrial ATP because it only works with oxygen. Okay? Very good. So,
Sir, what section are we in?
not sure. Catabolic means breaking something down. In biology, if you break big things into small things, you will essentially result in energy. You will make ATP. Okay? So if you right now break glucose into smaller things, you will get ATP. That process is called cellular respiration.
I'm good.
What type of process is this one? What do we call this one?
Hydrolysis.
Cellular respiration.
Wallah, I knew it. Quran, I knew it.
Why didn't you say it?
Because you said it before me.
You see, you must have O2 present to have a proper amount of ATP made by your body. Okay? You must have glucose reacting with the O2 to give you CO2 water along with lots and lots of ATP. This process is going to be exergonic because it gives you lots of ATP, which basically means you're going to be having a negative delta G. Energy now is being released to do active work. You must eat to, let's say, move or think or do something else. Now, if we have aerobic respiration, that basically means that oxygen is going to be consumed in the process. Okay? But if it's fermentation, that basically means there is no O2. And fermentation can happen in the cytoplasm. Okay, now if we're discussing a eukaryote, aerobic respiration will occur in two places in a eukaryote. In the cytoplasm and also in the mitochondria. Okay? So guys, look.
There is one too. I went out with him twice or once.
Let's talk about something called electron carriers. What do you think is the job of electron carriers?
Charges.
They carry electrons.
Is it like the electrolytic?
No, it's FAD and NAD plus. They will carry electrons, okay? And they will carry electrons, and after accepting electrons and hydrogens, they will become FAD H2 with NADH.
Oh, this is the Krebs cycle.
It's like they're found in there and also other things.
When are we gonna study it? Like the ETC? I don't know.
We're gonna study it today, hopefully.
Today?
Hopefully.
Yeah, calm down.
No.
Study what?
What unit is it?
Oh, I hate those.
Unit 3 right now. Which is yeah.
We're
So-
ahead of your class.
Hmm?
We're ahead of your class.
Yeah.
So can you close the recording, please? I don't forget.
So they will be carrying electrons, and at some point, these electrons.
So can we make bio lessons 1 hour 30 minutes?
If everyone here says yes, I don't mind.
Yes, no.
I think
So, so we say 6
Sorry,
from.
it's too long. Wallah, Baba, I get bored.
We say
All right, guys. So look, whenever I talk about carriers of electrons, I must also talk about electron transport chain. So ultimately, FADH2 and NADH both have to go inside ETC to basically to give away their electrons, okay, to produce ATP in the stage after.
No.
Okay. Now, where does the electron transport chain occur in the eukaryotes? It occurs on the inner
Membrane.
mitochondrial membrane.
Isn't this like the, wasn't there, I don't know if I'm wrong or not. There's like something and then Krebs cycle and then ETC.
Yeah, glycolysis,
Glycolysis, Krebs.
pyruvate decarboxylation, Krebs cycle,
Pyruvate.
ETC, then chemiosmosis. It's
So look, after
5
this, I
stages.
don't get it.
You see, guys, here we have the stages of cellular respiration.
What's that?
So
Okay.
It's in Rome.
Can I call?
I think this
Yeah.
is
I
wrong.
have to go one more.
But you already told us 740.
Just in case you forget.
We all heard you. Okay. He's
Yes.
calling someone else.
Has to make
Now, glycolysis.
See, sir, I told you it's 3.
It basically breaks glucose down. Then pyruvate oxidation, which basically means breaking down pyruvate, followed by the Krebs cycle, which can also be called the citric acid cycle. We also have finally the process called oxidative phosphorylation, which is made of 2 stages, the electron transport along with chemiosmosis.
So has this been added new?
No, it's always been there.
So what is this?
These are the stages to take you from 1 glucose to approximately 32 ATPs. They have to involve both cytoplasm and the mitochondria working with each other. Okay? Now, the first stage of cellular respiration is going to be called glycolysis, which is about breaking glucose down inside the cytoplasm. You must break glucose down into 2 pyruvate molecules. Now, glucose has how many carbons? 6. Okay? And each pyruvate has 3 carbons. Now, the pyruvate is not going to stay in the cytoplasm. It now has to move inside the mitochondrial matrix using a transport protein. And then it has to be made into acetyl-CoA, which will then enter the Krebs cycle, also called the citric acid cycle. Okay? You will then make CO2 by this cycle, which you guys will exhale. Right? Here's the entire process summarized. Some of it taking place in the cytoplasm, and some of it taking place inside the mitochondria. And of course, the amount of the ATP is going to be the highest produced by chemiosmosis, which is the last stage of these stages. So glucose becomes pyruvate. Pyruvate must then enter into the mitochondrial matrix to become acetyl-CoA, which will then enter into the Krebs cycle, and it will make you some ATP along with NADH and FADH2, which will then be taken up by the electron transport chain to give away their electrons to ultimately give you ATP by the process of chemiosmosis. So 1 glucose giving you approximately in total 32 ATPs, approximately. So in biology, the word oxidation basically means breaking something down. We have to break glucose down into pyruvate. A glucose is made of 6 carbons, but each pyruvate has 3 carbons in it. Now, for glycolysis to occur, you have to invest 2 ATPs. You have to put 2 ATPs to make glycolysis work to get 4 ATPs out of it. You will also get out of glycolysis 2 NADH, both of which are called electron carriers, along with 2 pyruvates, along with 4 ATPs out of just 2 ATPs. So glycolysis has to be powered up by ATP first for it to do its job. Okay? So you're gonna end up with 2 ATPs in net, 2 pyruvates, and also 2 NADH. This all takes place in the cytoplasm, whether there is O2 or no O2. That basically means both eukaryotes and prokaryotes can do this process, whether there is O2 or no O2. You see, whenever we have to use up ATP, we basically have to add a phosphate to our compound. So now if you use ATP here again, you must add one more phosphate to this compound. ATP is going to be essentially the source of phosphate, which will change the properties of that compound with the phosphate on it. So transferring phosphate from ATP is going to be a very important part of cellular respiration. It will turn glucose into a more active substance. Okay. And then we have another enzyme that can also transfer phosphate called phosphofructokinase. Whenever the enzyme's name, or whenever the name ends with ACE, it's going to be called an enzyme basically. So transferring phosphate from ATP is going to be a very important part for activating a certain enzyme. Now, if you want to block glycolysis, your best bet is to block phosphofructokinase, because it's the enzyme that ultimately controls glycolysis, right? So right now someone says, how can we shut down glycolysis? By shutting down phosphofructokinase. In fact, this enzyme is going to be called allosteric enzyme, meaning that it can be shut down by the binding of an allosteric inhibitor, which is called non-competitive inhibitor. Is the allosteric inhibitor. Now, once we have made pyruvate cannot stay in the cytoplasm. It has to make it to the mitochondrial matrix by being transported on a transport protein. Why? Because it's highly hydrophilic. I mean, look at its negative charge plus how many oxygens it has. There, it will be broken down by the enzyme called pyruvate dehydrogenase to make you CO2, to make you NADH, and also to make you acetyl-CoA, which is going to be the raw material for the Krebs cycle, or as the book calls it, the citric acid cycle. At this point, we know that the word oxidation means breaking something down. Breaking pyruvate down to acetyl-CoA is going to be very important to enter into the Krebs cycle.
So do I need to know those names?
Yes.
Why?
Why?
Yes.
It's required. It's required.
So I have to memorize all of this?
No, only what's in the blueprint.
So where'd you get the blueprint from?
He made it.
Would you believe me if I said that? If I said how?
No, okay. It's
How?
like-
Yalla.
Actually?
Yeah, go ahead.
ChatGPT.
So the word oxidation basically means breaking something down, breaking pyruvate down to acetyl-CoA, which then can enter the citric acid
I have a
cycle.
better idea. Why don't you?
So you're gonna get CO2 out,
seem like
2 NADH, electron carriers, ATP, FADH2 as well. So NADH and FADH2 are both called electron carriers. They will carry electrons. This is all happening inside the mitochondrial matrix, by the way, and you must have O2 for whatever happens in the mitochondria. So the next stage will occur in the inner mitochondrial membrane. This stage is called the electron transport chain, which will basically take the electrons of the NADH and the FADH2. It will remove their electrons. They have lots of electrons. And it will make them get carried on the inner membrane, which looks folded. So we're going to have electron carriers on this inner membrane, and they will take it from the initial point to the final point. They will get carried on carrier proteins until they ultimately reach point B, the final point.
The NADH is what carries them?
The NADH and the FADH2 will lose electrons. So all these electrons here are from NADH and also from FADH2
Where does this happen?
on the inner mitochondrial membrane. You see, guys, the NADH will give off electrons. FADH2 will also give off the electrons. This is all happening in the electron transport chain. All the electrons come from only two things, NADH and also FADH2. They have to be passed by complexes from 1 all the way to 4. What are they? They're simply proteins that can carry electrons. They act as electron carriers. Second. So the electrons get carried away from the NADH and also the FADH2. They will get carried on these complexes. This is all happening in the electron transport chain. And they have to be accepted ultimately. The electrons will be accepted by the oxygen. It will accept hydrogens and also electrons to give you water. So in that case, basically, oxygen is going to be the final electron acceptor in the electron transport chain. Now, during the electron transport chain, the hydrogen ions will get pumped into this space. This space is between the inner mitochondrial membrane and the outer mitochondrial membrane. This space has a name. It's called the intermembrane space. H pluses will accumulate in this space. Their accumulation here will create a lot of them in one area. When you have a lot of H's in one area, we call this the proton gradient. So a proton gradient is going to be created during the electron transport chain. Now, what comes after this is the last stage. The last stage is called chemiosmosis. In this stage, these H pluses now want to go back. They want to go back from this space back to the mitochondrial matrix. Because they are now in too much amount, or in too many amounts in one area.
All of them will go back?
Yes, all will now start to go back.
And then they'll be back with the same amount.
But look, the H plus is charged. So it's going to be hydrophilic. So it has to go back through a protein channel. And this channel is called ATP synthase. Not only it's a channel, it's also an enzyme because it ends with ACE. Now, as they go back through this channel, they will release energy. Sometimes the movement of things from large concentration to low concentration will release energy. This energy will then be used to make this enzyme work. And when the enzyme works, it will combine ADP with phosphate to give you ATP. So now what happens is ATP synthesis is going to occur on this enzyme. And again, this enzyme is only powered by the movement of the hydrogen ions from the area of high concentration to the area of low concentration. So ATP will be made in the last stage in a great amount. So whenever you see a proton gradient or a proton motive force, that's the result of the accumulation of H plus in one area, which is called the intermembrane space, resulting in an H plus gradient. What does gradient mean? It just means concentration of something in one area. And now when the H plus flows back through ATP synthase, this makes making of the ATP possible.
Because they release ATP?
No, because as they move back from high to low, this will now be used to release energy to be used for the enzyme to work. You see, the enzyme needs to mix ADP with P. but to do so, it has to use energy from somewhere. That somewhere is the movement of the H plus from high concentration to low concentration. By the way, mitochondria is not the only organelle that can make ATP by chemiosmosis. Chloroplasts can also do the same exact process. The name of it is called chemiosmosis, also using ATP synthase to make ATP. So chloroplasts do make ATP as part of photosynthesis. Now, as we can see in this figure, glycolysis will give us 2 pyruvates along with 2 ATPs along with 2 NADH. Now, then pyruvate oxidation will give you 2 NADH. Then the Krebs cycle will give you 6 NADH and 2 FADH2. Okay? And all the electron carriers, the NADH and the FADH2, will ultimately make it to the electron transport chain, where then they will dump their electrons, plus the H plus will move to one area, which will give you about 26 to 28 ATPs from chemiosmosis. You see, you only make 2 ATPs from glycolysis in net and 2 ATPs from the Krebs cycle in net. So you just make 4 ATPs basically before chemiosmosis. That means you make most of the ATPs during chemiosmosis. The ATPs that are made during chemiosmosis is because of the movement of the electrons. These electrons come from 2 electron carriers, NADH and also FADH2. If we see the word phosphorylation, does that mean removing a phosphate or adding a phosphate? Adding a phosphate. For example, when ADP mixes with a phosphate to give you ATP, that's called phosphorylation. Now, in case there was no oxygen, we have to stay in the cytoplasm. We cannot go to the mitochondria. This will be called fermentation or anaerobic respiration, which basically means your body will make you ATP without any oxygen, but that will only generate for you 2 ATPs, which is very, very bad for most eukaryotes. Okay. And this process will be without involving the mitochondria. So this process will be without cellular respiration, because respiration means O2 is there. There's going to be no O2. We call this anaerobic respiration, basically. So glycolysis has to happen when there is no oxygen. Okay. Look, if there is O2 or no O2, glycolysis will always happen, right? So right now there is no O2. If you guys see the word fermentation, that basically means there is no oxygen. Now, glycolysis occurs. It gives us 2 ATPs in net. It will also give us 2 NADH along with 2 pyruvate. Now, where does fermentation take place? So, if we're talking about a human being, it will take place in the muscles, right? Right now, if you overwork your muscle, it will get burnt. That's called lactic acid fermentation. But if we're discussing an organism like a yeast or a bacteria, they will make alcohol as a side product. So, the pyruvate that was made by glycolysis will lose a carbon to go from a 3-carbon substance to just a 2-carbon substance.
Why?
Why? Because we lost a carbon as CO2. That means organisms that perform alcohol fermentation will also produce CO2 as a result. Okay. Now, what's the point of fermentation? Why can we just not let glycolysis take place? Why fermentation? Because fermentation makes sure to turn NADH to NAD+. Because without NAD+, glycolysis cannot occur. Okay? So, you see, glycolysis needs a couple of things to start working. It has to have 2 ATPs. It has to have a glucose along with 2 NAD+. And fermentation is the process that will give us a constant supply of these 2 NAD+. And by doing so, glycolysis can now happen permanently without having any O2. That means for every glucose, you will get 2 ATPs in net. So for every glucose, you will get 2 ATPs in net, you get 2 pyruvate, and you get...
4 ATPs.
No, because we used up 2 to make 4. But net will be
What's net?
the outcome minus the income.
Oh.
Yeah. So the net ATP will be 2. So guys, if we have anaerobic respiration, you will only have glycolysis and fermentation. There is going to be nothing in the mitochondria, and both will take place in the cytoplasm. So glycolysis will give you 2 ATPs in net. Fermentation gives you 0 ATPs. The whole purpose of fermentation is to keep glycolysis occurring by always giving it NAD+. That's the only job of fermentation, to keep supplying us with NAD+ for glycolysis to keep occurring. And this fermentation happens in humans, lactic acid fermentation during workouts, for example, and you will turn a pyruvate into lactate or lactic acid, which will cause muscle ache. So any muscle ache after a gym is because of lactic acid, basically. That's burning through the area. But basically, both fermentations will give you 0 ATPs. Glycolysis is the one that gives you 2 ATPs in net. You see here, after glycolysis takes place, the pyruvate will have 2 fates. Either no O2, which means fermentation, or O2, which basically means we go to the mitochondria and be used to make other things. If you go to the mitochondria, you make about 32 ATPs. If you stay in the cytoplasm, you will just make 2 ATPs. So what we see here is a process of negative feedback, where basically, once your body made enough ATP or made enough citrate, it will go back and it will tell this enzyme, which controls glycolysis, to stop working. And this will shut down the entire process. So guys, basically phosphofructokinase is the enzyme that can regulate cellular respiration. If you want to stop cellular respiration, simply stop this enzyme from doing its job. You will stop it by inhibitor. That's why it's going to be called allosteric enzyme, because the inhibitor will bind on its allosteric site, basically. Okay. So guys, look, we love plants, right? Because plants give us food. Yes, they make food for themselves. We come and we take their food, basically. So that's the idea of photosynthesis. Using light from the sun to make food. Okay, because they have a chloroplast. Okay, now autotrophs are going to be organisms that can make their own food, whether there is light or no light. So for example, there is this type of bacteria that can make their own food without using any light. They can also qualify as autotrophs. What are some examples of autotrophs? Algae, plants, and some bacteria. Some can do photosynthesis, and some can do something else called chemosynthesis, which means without any light. If bacteria can do photosynthesis, we call them cyanobacteria. And if they can do food making without light, it's because they use sulfur, and they have to live in the deep oceans, basically.